In our lab, our group was able to successfully extract the strawberry DNA from the cell, and this is how we did it. The materials used in this experiment include 1 Ziploc bag, 1 frozen strawberry, 20 milliliters of detergent solution, 1 graduated cylinder, ethanol that was chilled, a coffee filter, salt, meat tenderizer, a test tube, a stirring rod, and goggles. As you can see, we have Nate crushing the frozen strawberry in the Ziploc bag. By doing this, Nate is breaking down the cell wall, releasing the nuclei where the strawberry DNA is. After squeezing and smashing the strawberries for 2 minutes, we started to add our other ingredients. We started to add our 10 milliliters of detergent solution, the meat tenderizer, and the salt. Groups in the class discovered that 10 milliliters wasn't quite enough, so Miss DeBaney instructed us to add another 10 milliliters of the same solution to our strawberry mixture. Nate is now demonstrating how we released any extra air in our Ziploc bag, resealed that bag, and then proceeded to squeeze the strawberry mixture for another 1 to 2 minutes. The soap in the detergent breaks down the fatty membranes of the cells. It breaks open the nuclear membrane and releases the DNA into the solution. The salt in the strawberry mixture makes the DNA molecules within the strawberry stick together and separate from the proteins that are also released from the cells. During the experiment, our group found it very hard not to puncture holes in the bags while mashing the strawberry mixture. There were multiple incidents in which strawberry had come out of the bag and onto the table or flying at people. So it is very important to take extreme precaution when handling the Ziploc bag and smashing the strawberry mixture. Now we have Ricky placing the filter into the funnel. In our first attempt of pouring the strawberry mixture, we let it drip into the graduated cylinder till it was about 1/8 of the way full. To speed up the process, Nate squeezed the wet pulp to release any more liquid. In doing so, he also released a little bit too much of the pulp from squeezing it too hard. We then had to restart our funnel setup and re-pour the liquid into the Ziploc bag. Repeating the following steps from earlier, just not as aggressively. Ensuring that the filter removed all large particles from the solution, such as seeds, pith, etc., allowing only small cells like DNA and proteins to filter through the liquid. Then pouring our strawberry solution into the test tube, we also placed a layer of the cold ethanol into the same test tube. As you can see, the immediate results were that of the two liquids separating into two layers. The DNA precipitating out of the strawberry solution between the layer of alcohol on top and the extract on the bottom. Taking the stirring rod, placing where the layers meet, then pulling the rod up and stirring it around, we found the long thread-like DNA molecules that were a whitish, gooey, and stringy consistency. This is the DNA containing strawberry genes, containing all the proteins needed for the strawberry. In this experiment, we successfully extracted the DNA from the strawberry. We could clearly see the DNA clumps and strands. By first breaking down the cell wall through crushing the strawberry, then adding the salt, detergent, and meat tenderizer, they dissolved the cell and nuclear membrane. We then separated large pulps by filtration. Some controls we kept throughout the experiment were measurements of the liquids and our time. We would extract DNA from multiple fruits to see which extracts the most DNA cells through fruits with different chromosomes. We would also be more careful with our Ziploc bag.